Booyah. All right, continuing to talk about speciation. Let me just give you an example of what I was talking about earlier of how coming up with good species definitions gets messy. This is a California wildflower. The common name of it is farewell to spring. It used to be called Clarkia deflexa. It's now apparently been renamed. It's Clarkia bote. Let me see, check the chat. We've got, question was, will there be any calculation problems on the test? The answer is no, you do not need to bring a calculator. So Clarkia is a common Southern California wildflower. And in the 1950s, Harlan Lewis collected Clarkia from 11 different populations in Southern California and experimentally started crossbreeding them. He didn't manage to crossbreed each of the 11 with each of the other 11, or each of the other 10. So the data is not complete, but it still shows some pretty interesting results. So that's a range in green of, that's the range of Clarkia deflexa. The 11 populations are A through K, going more or less north-south, from A through K. And there's his original data, and it's a little hard to read, so I've color-coded it for you. The green lines link populations that were completely interfertile all the way to F2. So C and J, for example, he crossbred those, he got fertile offspring. He crossed those fertile offspring, and the offspring of that offspring were fertile. So green is complete interfertility for at least 2 generations. So C and J and C and F and D and E and D and I were completely interfertile. Already you can notice you've got some interesting problems because D and I were interfertile, D and E were interfertile, but E and I are not interfertile. What's going on here? In yellow, he crossed those linked populations that he crossbred, and the F1 offspring were fertile, but they couldn't produce an F2. The F2s either died or the F2s were sterile. And then red indicates that F1 either died or was sterile. And then the thin black lines are cases where no F1 generation was even produced, no interbreeding at all. So A is pretty clearly not at all interfertile with any of the populations it was tested with. All those red lines are populations where there was either no F1, either the F1 died or the F1 was sterile. And so you can see we've got everything from complete interfertility to complete intersterility. But aside from population A, which was isolated from everybody, it's not easy to see how to divide this up. So you've got, D and I can fully interbreed, D and E can fully interbreed, but E and I can't. So you could get genes from E into I, but they would have to pass through D. Let's see. I mean, you have C and F can interbreed. And then J, I guess he did manage to test. But you have like C cannot interbreed with D. D is fully interfertile with E. E is not fully fertile with C. You've got all these combinations, and it's not at all clear if there's any way you can divide these populations up into separate species, except you could argue A probably is. As for the rest, you've just got a mess. Now, in part to handle some of these difficulties, there have been some alternative attempts to come up with a good definition for species that address some of the deficiencies in the biological species concept. The morphological species concept is basically what Linnaeus was doing, and it's basically what your Audubon Society bird books do. They define a species by a unique feature that it's got, some unique physical feature that they have. The problem is that, first of all, as we've seen, you can have cryptic species that may look identical but be completely unable to interbreed. It's also true that some of those morphological features can be extremely hard to go out and measure. If you ever have hung out with the Adamses very much, you might have found that to identify certain species of small freshwater fish, you have to do things like count the number of scales in a ring around the caudal peduncle or something like that. Finding features that separate species out can be a very subtle business indeed. And you can also have just species that are very variable and yet all can interbreed. Ecological species are defined by the niche that they inhabit. So even if two of our Clarkia species can potentially interbreed, if they're in different niches, then we call them separate species. And the behavioral species concept defines a species by what they call its SMRS, and that's a specific mate recognition strategy. The behavioral species concept does note that many species have very different senses than we do. So you might have two moths, let's say, that look identical, but looks are not what's important when you're a moth. Smell is. Moths have, I don't know if you saw it on that hawk moth yesterday, but did you notice they've got big feathery antennas? Yes? Okay. Big feathery moth antennas are big feathery chemosense organs. Moths can home in on the source of single molecules of moth pheromone. So in that case, it really does not make any difference what the moth looks like. That's not important to defining what species are. What's important from the moth's point of view, or not view, the moth's point of smell, is going to be the pheromones, and those would be the specific mate recognition strategy. Those meadow larks that I showed you earlier, you can't tell the eastern and the western apart, but the males sing very different songs. So in bird world, it's not necessarily what you look like, although it could be, but it could be that what's most important for defining species is not how you look, but how you sound. Are any of these much of an improvement? Here's a classical study by a man named Albert Blair. He went around studying North American toads. You know, Conway's official animal. Right? Yes? You have toad suck days. My kid was 4 and we actually hunted down a toad and entered it in the toad races. And it came last and then promptly died the next day. So, oh well, so it goes. So he went around studying toads, and I'm just giving you a taste of his total findings. He went around collecting toads that he assigned to one of 2 species, Bufo americanus and Bufo fowleri. And that's what they look like. There's a fowleri on the left and an americanus on the right. And yes, they do look very, very, very similar. Are they one species or 2? As you will see, the answer turns out to be a very loud and resounding, Write that down, UNH, that's gonna be on the test. I'm gonna test you on, and he crossed, in the little snippet I'm showing you, he took female Bufo fowleri from Indiana and crossing them with male Bufo americanus from elsewhere. And there's some of his data. When he took his Bufo fowleri and crossed them with male americanus from Bloomington, Indiana, the percentage of eggs that yielded tadpoles ranged from 40 to 95. When he collected male americanus from a different lake in Indiana, he got nothing. And then when he collected a bunch of toads from Tulsa and crossed them with his Indiana females, he got everything from almost nothing to almost everything. From 4% of the eggs yielding tadpoles to 95% of the eggs yielding tadpoles. So that's our problem with the biological species concept, that we've got a mess. We've got everything from complete interfertility to complete intersterility. And it's not in such a way that we can neatly divide up our toads into separate species. So we're hybridizing what we used to call two separate species, and we get everything. We get an answer of, Are they separate species? Well, if you go by the behavioral species, they probably are, because Americanus breeds at shallower ponds, it breeds earlier in the year, and the mating calls are quite different. And that's often important in animals that find mates by sound. Birds and frogs often use very specific songs or calls or wavelengths to attract females. And so that's an important part of the specific mate recognition strategy. So Americanus and fowleri probably are separate species if we go behavioral. Morphologically, they seem to be the same. Americanus can look a little different, but it's not consistent enough that you can identify one by sight. Ecologically, they're not the same species because they overlap in their niches a great deal. There may be slight differences, but they mostly share the same niche. Biological species concept, we get, because we get, we can cross two populations and get everything from complete sterility to complete fertility. And what we now probably do as working scientists is simply pump the ball and call this an example of a species complex. A species complex is a whole bunch of species that are in practice very hard to distinguish, and somebody else should really do the work of trying to figure out what the real separate species are, and somebody else can do that. We're not going to. There's a common mushroom around here. I've actually seen it growing in my yard, and it is theoretically edible. And it's supposed to be delicious. I've never been tempted to eat it because it belongs in the genus Amanita. And Amanita also includes the mushrooms that will kill you in a very painful, agonizing way over the course of a week. So I see this mushroom and I figure there's a 99.9% chance that if I eat it, it'll be delicious and I'll be fine. But there's still a 0.1% chance that if I eat it, the only possible treatment is going to be liver transplant. And I'm still using my liver. Anyway, so I don't eat it. But it used to be called Amanita jacksoni. Now it is generally recognized that what we used to call Amanita jacksoni is a whole bunch of populations that are an absolute beast to try to tell apart. So this is usually called a species complex, and it's usually called Amanita section Caesariae stirps hemibatha. And within that, there's a whole bunch of species that nobody really has figured out how to tell apart. And so we basically just go, yes.
So all of these concepts, they don't take into account the genome, like gene sequencing.
They were developed before DNA sequencing became really a thing. But I mean, DNA doesn't necessarily make things easier. I mean, you could go sequence a bunch of organisms and find that they've all got different DNA. But is that DNA different enough that we should call them separate species or not? I mean, there's nothing about DNA that says, hi there, I'm Anita Jackson and not something else.
So there's like, it's hard to say if they're close enough or they're different.
Yeah, well, it can sometimes identify, it can often identify introgression. You know, it can say DNA from population X and population Y shows that there is some gene flow between population X and population Y. Is that enough to make them separate species? Well, that's where you get into territory. That's where you get things like polar bears and grizzly bears that really look like they ought to be separate species, but actually have been interbreeding to some extent for a long time. So it's not that DNA is irrelevant, it's just DNA does not magically give you more clear answers. DNA can just say, okay, we have two individuals and they are different and such a way. It doesn't give you a yes, no answer to the question, are they separate species or not? And again, if this sounds messy, that's exactly what we would expect to find if evolution really is taking place. You should always have species that are coming into existence but not fully separated, or maybe that were partially separated but now are coming back together, or are beginning to separate but not quite, or mostly separated but they're still exchanging genes. No matter what species concept is in use, this kind of ambiguity is what you should expect. And dipping into history for just a moment, I want to talk about something Ernst Mayr, as an old man and a historian and philosopher of biology, pointed out as being fundamental. We have a shift from typological thinking to population thinking. And we'll start with Linnaeus. Carl Linnaeus was a Swedish physician who had an ego that would not fit through double doors. He literally saw himself as being on a divine mission from God, as they say in the Blues Brothers, in that Chicago accent. They're on a mission from God. And Linnaeus was on a mission from God to study the natural world and classify all of it. And as we saw way back in the Day the Universe Changed video, he felt that if he could just understand the right way to classify everything, he'd be getting inside God's head. He would have some sort of new insight into what God had been thinking. And that's an actual quote. Linnaeus is speaking of himself in the third person here. God has allowed him to see more of his created work than any mortal before him, right? Modesty was not a problem for Linnaeus. Low self-esteem was not something that seems to have bothered him. God has endowed him with the greatest insight into natural knowledge, greater than any has ever gained. Think well of ourselves, do we? And I know you remember the taxonomic hierarchy, right? Domain, kingdom, phylum, class, order, family, genus, species, right? Do kings play chess on fat green stools? Yes. Did King Peter come over from Germany swiftly? Do kinky perverts come out for good sex? right? You have lots of ways you can memorize that taxonomic hierarchy. And I know that last one was a little gross, but it ain't nothing compared to the way that medical students memorize body parts. Yeah, the mnemonics for memorizing the 12 cranial nerves can get a little bit rough. Ditto for the wrist bones. Anyway, Linnaeus went back to the Greek philosopher Aristotle. There's Aristotle. And he classified things. He said, okay, if we want to classify things, we should first figure out what they all have in common. And then we should figure out what makes each one of the members of the group different from each other. We first need to figure out what all members of the group share. And then we have to think the way they used to on the old Sesame Street when I was a kid, and think one of these things is not like the others. Anybody remember that? Yeah, they had this Sesame Street bit where they'd show you like 3 apples and a rubber ducky and sing this song. One of these things is not like the others. One of these things just doesn't belong. Can you guess which thing is not like the others before I finish my song? And then you'd have to figure out that in fact the rubber ducky was different from the apples or whatever it was. And I can't remember the Greek words that Aristotle used, but his word for, the features that all members have in common was genus. So Aristotle's genus is humanity, because all members of humanity have 2 eyes and a nose and 2 ears and so on. What makes Aristotle different from the others is he's got a bald spot, he's got a comb over, he's got a big nose, he's got a beard, he goes around philosophizing. He used to be Alexander the Great's tutor before Alexander the Great decided that conquering the world was better than studying it. So Aristotle has all these things that make him a specific member of the generic group humanity, or man, as you like. Linnaeus was the guy who formalized this into the taxonomic hierarchy, that a species is, a biological form that has at least one thing that makes it different from all other species. But a group of species that are similar in most respects, all those similarities make up the genus. And then, of course, genera. Originally, there weren't families. He put genera into orders and orders into classes, and people later added phylums. And he published all this in a book called Systema Naturae. So here, this is a page for it. His first order of mammals, that's ordo up there, is primates, which he defines as dentes primores superiores quatuor paralleli mammae pectorales vinae. Upper front teeth are 4 and they're parallel. So 4 upper incisors that are straight. Nipples on the chest and in pairs. Instead of having rows of nipples like a dog or a cat or an udder like a cow, primates have 2 nipples and they're on the chest, not anywhere else. So his first genus there is Homo, which he just describes as nosce te ipsum, recognize yourself. Or in other words, I'm talking about you here. And the first species, this is an earlier edition, and his first species, his earliest species name was Homo diurnus, man of the day. And down below, you might be able to see Homo nocturnus, man of the night, which is what we now call the orangutan, and which is now in a different genus, genus Pongo. He's divided, by the way, Homo diurnus into Homo rufus, red man, Homo albus, white man, Homo luridus, yellow man, and Homo niger, black man. And then finally, Homo monstrosus, which is where he puts all of these strange travelers' reports of giants and dwarves and things like that. So that's his early draft of how you classify things. And the 10th edition of his book in 1758 was the first to use binomial names throughout. That's the starting point for biological taxonomy. Any Latin names that were published before the 10th edition of Systema Naturae are not used. And a number of his Latin names are still in use today. Bos taurus for the domestic cow, Homo sapiens for us, Canis familiaris for the domestic dog. Most of the very familiar domestic animals still retain names that were given by Linnaeus. Of course, we've found a lot more species since then. Linnaeus never doubted biblical creation. He did eventually suggest that there might have been some new species would come into existence after creation by hybridization. As we'll see, sometimes if you crossbreed two species of plant, you can get a third that doesn't look like either parent. That can happen, and Linnaeus noticed it. But aside from that, each species must have been created separately in the Garden of Eden. He suggested that the Garden of Eden must actually have been a huge mountain, because a mountain is the only way you could get Arctic species and tropical species in the same limited area. If you put the polar bears on top and the capybaras on the bottom, is the only way you can get tropics and Arctics in the same place. As he was to say, the earth's creation is the glory of God as seen from the works of nature by man alone. Reactiones telluris est gloria Dei ex opere naturae per hominem solum. And species couldn't vary, or if they did, it was irrelevant. It might be true that all members of the species might look a little different from each other, but what's important in this kind of scheme, if you're trying to work out what real species are, because you believe that they are all individual expressions of God's thought, and that classifying in the right way will let you see how God is thinking. If you want to get inside God's head, you define each species by some sort of intrinsic nature that God had defined. That all members of Homo sapiens are members of that species, ultimately because they share some kind of divine essence, some kind of humanity nature that God put there. All dogs are dogs, not because they look alike, but because they all have some sort of essential innate dogness, dogosity.
Dogometer.
What's that?
Dogometer.
Okay, you can measure it with a dogometer or something like that, but they've all got some sort of inherent dogness, and it's your job to figure out what that is. As he said, the invariability of species is the condition for order. And there are species that vary a lot. As I said before, this is a Cuban land snail called Polymyta picta. And you can see that the shape and the color and the banding pattern of all of these is very variable. And it's not obvious how you could divide this into subtypes, because there isn't a variant there that doesn't share features with some other variant that's otherwise different. But to Linnaeus, that's all irrelevant. What's important is this true nature, this true Cuban land snailiosity that God put there. and everything else is a distraction. And this still is maintained in biology because it's implicit in the way that we define new species. If you were to go out to the Brazilian rainforest or the wilderness of the Ozarks or wherever, and go out and study the animals and plants and fungi and everything else, you might eventually find a new species. And in fact, there are something like 10,000 new species discovered each year. We've probably found almost all of the big photogenic mammals that are out there, but we keep finding new insects and spiders and plants and fungi and all kinds of things. And if you find something that you think is new, let's say you find a plant that doesn't look like anything you've seen before and you collect it, you first go around and consult all of the original descriptions of similar species. And if you think your new plant is different from all of them, you write up an official description of your new species. And in plants, by the way, you actually have to publish a description of your new species in Latin. It's one of the few areas where you still have to compose in the Latin language. Although botanical Latin is such a limited language that it's actually not that hard because there's a limited number of things you ever have to say. Hello. Hi, you looking for something or?
Just looking for classes.
Okay. You doing what, 1440 with Carafit
Yes.
this fall?
Yes.
Okay.
Is that, are you,
I'm not Carafit. This is a different class in the summer, but I am told he's going to be teaching here.
awesome?
Yeah. Okay. Tell him to bring his guitars in sometime. He's yeah, he's a good biologist. He's also a pretty good musician. Yeah, if you need to distract him, then that's a good thing to get him. He actually builds, you ever seen a cigar box guitar? Yeah. It's a type of guitar that you can make from things lying around the house. It's got a cigar box for a body, and like you could use a fence post for the neck. He makes those. Yeah, get him talking about that, and maybe he'll be so distracted, you won't have to learn the hard stuff.
That's a whole lesson, just like that.
Well, let's not get too optimistic here, but yeah, he's a good guy and he knows his stuff.
Awesome. Awesome. Trying to start me.
No, carry on.
Thanks for the tip.
Poor Steve. Right. Okay. So where were we? So this typological thinking, you find what you think is a new species of plant, you study the heck out of it, you describe it very precisely, you compare it with descriptions of everything that might be like it. If you still think it's a new species, if it doesn't match anything that's already known, you write an official description of the new species. And if it's plants, you still have to write your description in Latin, although you'll also write a fuller description in English or whatever your native language is. And you have to deposit what's called a type specimen. This is a preserved, a pressed plant, and this happens to be the type specimen of some Brazilian flower. And the type specimens are deposited in museums, usually in very carefully preserved collections that are not open to the general public. There's a lot of museums. You think of a museum as something that's meant to, entertain and inform the public, but there's a lot of museums in which the collection is not open to the public at all. And there might just be minimal public exhibits or maybe even none. When I was in grad school, I worked at the University of California Museum of Paleontology, and we did have a few exhibit cases and things like that. And in the lobby of our building, there was a T-Rex skeleton mount. But the vast majority of what we had was not on public display and wouldn't have looked all that interesting to most people. But we did have a whole room of type specimens, which I couldn't even get into most of the time. You had to keep those types very securely, because the type is forever the standard reference for defining what this species is. I can't even read the name of what this is, but this is the type of a Brazilian flower. It's kept in Herbarium Hortensis Cuensis, the herbarium collection of the Royal Botanical Gardens at Kew, KEW, in South London. Kew Gardens, by the way, are this really great botanical garden with these magnificent old greenhouses made out of wrought iron. So you've got these great tropical plants in the middle of London. Kew was the palace where King George III was kept when he went crazy. So Kew was where he was when he went running around having screaming arguments with the trees. But I digress. They've also got a big collection of preserved plants. And if you're ever studying this genus, you might have to go over to London and examine the type specimen directly, because this is supposed to be the standard reference for defining what that species is. We don't do it this way now, but there was a time when the official definition of what a meter was the difference between two marks engraved on a platinum bar that was kept in a vault in Paris. There is like one official, bar of platinum alloy and two marks engraved on that bar, and that is the standard of the meter. And if there's ever any doubt whether like your ruler really is a meter long, in theory, you could go to that Paris vault and directly compare it with the standard. This is not how we define scientific units now, by the way, but it's the way we did for a couple hundred years. And we still do it this way to an extent in biology. You also still get this typological thinking in the thought of modern young Earth creationists. This is part of the statement of belief of the Creation Research Society. And to be a full member, you basically have to sign on to this. And part of their statement of belief is that all basic types of living things were made by direct creative acts of God during the creation week described in Genesis. Whatever biological changes have occurred since creation week have accomplished only changes within the original created kinds. You will get this a lot. You will get arguments like, okay, evolution can make changes happen within a created kind, but it can't, it can't create any new created kinds. And then if you ask them to define exactly what they mean by a kind, they will generally change the subject as fast as they can, because they're not entirely sure what it is, but they know it when they see it. And this is very typological thinking. This is basically carrying on the tradition of Linnaeus and the way he thought. That all of the fundamental types of living things were separately created, and variation might happen within those boundaries, but it can never cross them. Linnaeus's classification is actually an awful lot like classifying chemical elements. How do we classify elements?
Number.
Okay, what's the scheme that we use? Like when you arrange a bunch of elements on one sheet of paper, how would you do it? Okay, what's the diagram that we use? Yeah. So if you think about the periodic table, it's got a limited number of squares in it, right? And every possible atom is supposed to fit into one of those squares, right? You define that by atomic number, right? That's the number of protons in the nucleus, and also the number of electrons in an uncharged atom, right? Yes. And so every atom can be put into its own little pigeonhole. If you have an atom with 6 protons in the nucleus, it's carbon and it goes right here. If you have an atom with 92 protons in the nucleus, it's uranium and it goes right here. And then you can go further and subdivide those little boxes based on the number of neutrons. So you're talking about different isotopes and so on. But there is a place for every atom. And even if we didn't, even if we haven't discovered a particular element, we can still place it in the periodic table. For a long time, there was a hole at, I think, element 43, but it was possible to predict what its properties were, and eventually somebody figured it out. It's a radioactive element called technetium. There's all those elements, all of those extremely unstable elements that have, we've only been able to create a few atoms for a very short length of time, but there's a space for them. Before anybody was able to make an atom of octium, we knew there'd be a place for it. Right? Yes? Okay. So there's a pigeonhole for everything, a place for everything, and there's no way you can ever have an atom that sort of has 6 protons in the nucleus, but maybe kind of has 7, right? You're never going to find an element whose atomic number is 6.5, right? You're never going to find an atom that is kind of like carbon, but also sort of like nitrogen. Yes? Every possible atom goes into a pre-existing pigeonhole on the periodic table. And of course, this results from the way that protons and neutrons build nuclei. And then you can get deeper into the periodic structure and start looking at verticals and start looking at, halogens and alkali metals and so on. But it all ultimately comes down to, the way that subatomic particles fit together. And it's not the sort of thing that is ever going to change. Yes.
Oh, I just wanted to say it sounds sort of silly. Great Creation Week sounds like a event on par with Shark Week.
Oh, Creation Week is kind of like Shark Week. Okay.
And it's kind of silly, like sort of interposing.
Right.
The direct creative acts of God during the Shark Week.
We've heard about the time God was talking with the Archangel Gabriel, and God is saying, I had a really good day today because I came up with this concept, and then we've got this new stuff called light. Gabriel said, that sounds really cool. God said, and then I started thinking about what's the opposite of light. And I came up with this thing called darkness. Gabriel said, that's pretty amazing. God said, well, it's even better. In this world that I'm creating, I'm going to fix it so there's light for a certain length of time, and then followed by darkness so that people don't get bored, right? We're going to have light and dark alternating. And Gabriel said, that sounds really cool. God, what are you going to do now? And God said, oh, I'm going to call it a day. Get it? All right, waka waka. Okay. Here's the thing, and here's why the Creation Research Society isn't so much wrong as they are 300 years out of date. In an evolutionary sense, variation is critical. To Linnaeus, variation just got in the way of your understanding of what the true essence of a species is. To an evolutionary biologist, variation is the raw material that makes evolution possible. Species can change, species do change, species vary, These are all specimens of a moth from southern Arizona. They're all the same species, but you can see how they vary in wing pattern and in color. Variation is the core. Variation is utterly fundamental to evolution. And sometimes that variation slops over the edges of your nice, neat definitions. If classifying atoms worked like classifying species, you could have an element that was kind of in between carbon and nitrogen and had an atomic number of 6 and a little bit. You know, you could have atoms that could only be classified as and you certainly do have that going on in biology. Variation is everything. And Darwin went so far as to suggest that species should not be considered to be real. Species were convenience that people came up with. Naming species would be a little bit like naming colors, in that it's convenient to talk about red, orange, yellow, green, blue, purple, etc. But in reality, those are all just points along the spectrum, right? There's no, if you see the full spectrum of visible light, there's no dividing line between red and orange. Red just kind of fades into orange. Some cultures don't even bother with a word for orange. They've just got a word for red and a word for yellow. Russian doesn't have a word for blue. It's got a word for light blue and a word for dark blue, but no word that corresponds to what we call blue. And so our words for color are arbitrary. You know, it's convenient to divide up that spectrum, but the truth is that all you really have is a spectrum of visible light, wavelength between 300 and 700 nanometers. And any kinds of colors that we block off in that spectrum are for our convenience, but they don't really correspond to anything, you know, unique and real. Darwin says species should be treated the same way. The only distinction between species and well-marked varieties is that the latter are known to be connected at the present day by intermediate gradations, whereas species were formerly thus connected. We shall have to treat species in the same manner as those naturalists treat genera who admit that genera are merely artificial combinations made for convenience. This may not be a cheering prospect, but we shall at least be freed from the vain search for the undiscovered and undiscoverable essence of the term species. That's the shift from typological thinking to population thinking that has been identified as such an important turning point in biology by Ernst Mayr. So these are very much like seashells that I used to pick up on the beach when I was a little kid. And we took our vacations at Fort Walton Beach, Florida. This is a little surf clam called Donax variabilis, AKA the coquina. And you can see it's very variable in its patterning and its color. And there's no really obvious way you could subdivide it into separate species because all of the different varieties just kind of blend in with each other. And that's what we mean by population thinking. Nobody today is going to try to figure out what is the true essence of Donax variabilis. What they would study is how it varies and how it might fade in with other species of clam. You know, you have to be used to confronting that messiness, that factor. And I'll tie this off by introducing somebody that's actually fairly relevant. This guy's an entomologist by the name of Alfred Kinsey. Kinsey is more famous for his other work, which I'll talk about in a minute. But he was an expert on a family of wasps called the Cynipidae. Cynipid wasps are very small. You're very unlikely to ever see them unless you're lucky or you really go looking for them. They don't sting. And female cynipids lay their eggs in plant tissues. In fact, true cynipidae are almost exclusively lay their eggs on oaks. When they do that, the plant is stimulated to start dividing cells. And the plant forms this swelling around the egg called a gall. And when the eggs hatch, the insects eat the gall from the inside out and eventually pupate and then come bursting out of the gall. It's not too hard to find these in the nature reserve, especially because they last a long time. If you just look at the dead leaves under an oak tree, you're likely to find a dead leaf with a big round swelling on it that if the leaf is dead, this will be dead too. It will be brown and dry and hollow on the inside and about the size of a golf ball. That is the gall of a cynipid wasp. This is the kind of thing that Kinsey was the world expert in back about 100 years ago. There's lots of other insects that can produce galls, and they're not always wasps. There are aphids, for example, that make galls on sumac that you can see right now. But Kinsey was a world expert on cynipids. And he drove around every summer in his Model T Ford with a couple of grad students who couldn't get away in time, collecting cynipid specimens from all over the United States, going from oak grove to oak grove, collecting his cynipids and trying to figure out what cynipid species really were. As he pointed out, too many systematists, that's scientists who study classification, attain their objectives when each species is represented by a half dozen specimens pinned in their cabinets. Too many people would collect maybe 5 or 6 wasps, say, hey, these look different, let's call it a new species, and that's it. These are the systematists responsible for the definition of systematic entomology as the science of transferring pins from one box to another. So most people were defining species, but not really coming to grips with variation. And Kinsey went out and sampled like crazy and collected over 71,000 cynipids to try to figure out what are the real limits of variation. How variable are our cynipids? He concluded that things could get fuzzy, but it was still useful to define species as real, and he compared it a lot to defining rivers and streams. Streams can flow together, streams can split. Sometimes if a stream flows into a marsh, it can be really arbitrary to figure out where the stream really is. If it's flowing through wetlands where you can't really define a single stream course. And streams are, of course, always changing, always eroding banks and building up sandbars and changing flow levels and currents and all that. It's still useful to try to define streams, even if you can never truly pin it down. That we can still talk about populations that tend to go together as species, even if you can never absolutely decide where the boundaries lie. That speciation was still useful. By the way, he put this in probably his 3rd most famous book, the gall wasp genus Cynips in 1929. Well worth a read if you're into wasps. I've actually used it in research before. Here's the other thing that Kinsey's famous for, by the way, probably more so than Cynips. Biology departments used to offer classes in what was usually called a class in family life or something like that, but it was basically sex ed. One like this was still being offered as a non-majors option at Tulane when I was there. These days, you can find out more about sex than your grandma ever knew by a simple matter of, 30 minutes on Pornhub or something like that. But, time was that, good people never learned about that sort of thing. And there would be college classes in marriage and family life. And like biology professors everywhere, Kinsey had to teach non-majors courses, including the biology part of a course in marriage and family life. So he basically had to teach sex ed to a bunch of Indiana farm kids. And he tried to do a good job. And he tried to do some research on human sexual behavior. And he found out there were a lot of references to abnormal sexual behavior. There were whole books out, books that were generally written in Latin so that only a few scholars could understand what the books were about. You wouldn't want kids getting in there and reading about all of this strange, psychological polymorphous perversion. So there was data out there about the extremes, about the fetishists and the sex maniacs, but there wasn't any data on what ordinary people actually did. There was no real baseline for deciding what was normal and what was abnormal anyway. And Kinsey applied the same approach to human sexual behavior that he did with his gall wasps. Instead of arbitrarily defining a standard of normal and trying and ignoring the variation, or trying to, treat the variation as freakish, you had to make lots and lots of observations and try to figure out what the total range of variation was. You can't figure out human sexuality by talking to 5 or 6 weird guys in trench coats. You've got to measure the full spectrum. And Kinsey started sending out surveys on sexual behavior all over the place. And after World War II, he published 2 books called Sexual Behavior in the Human Male and Sexual Behavior in the Human Female. And Congress tried to have him fired. They were enormously controversial at the time because they blatantly contradicted a lot of the things that we would like to believe about ourselves. Kinsey found that roughly 1 in 10 human men had some same-sex experiences. You have to, this is still sometimes quoted as 1 in 10 of 1 in 10 men is gay. That's not quite right, because you have to be very careful with how you phrase questions like that. If you ask the question, do you exclusively gain sexual pleasure from men, you will get a very different answer than if you ever ask the question, have you ever looked at Jason Momoa and kind of wondered, right? Very, very different. Yeah, like I said, it depends on how you phrase the question, and it's not easy to write surveys like this that are where the data is accurate. But he found that 1 in 10 men had at least some same-sex leanings. Found out that in rural Indiana, there was a surprising number of men who had sexual experience with farm animals. Somewhere, he was not noted for a sense of humor, but he supposedly quipped that according to his surveys, 3/4 of Indiana men should be in prison and the other quarter were anemic. And it really hit up when he published Sexual Behavior of the Human Female and realized that premarital sex was not much less common than it is now. Your grandmas were not necessarily flowers of unblemished virgin purity until marriage. There was plenty of sexual experimentation going on. Again, not much less than today. And that's what almost got him fired, because people were claiming he was a communist who was undermining America's morals and undermining the purity of its women, who have always been, God-fearing, proper ladies who would never dream of such a thing except for, those debased, painted Jezebels and shameless wenches. But real American women were always pure until they got married and had lots of kids and baked them apple pie. I'm proud to be an American. Sorry, but you get the idea. And yeah, he was thought to be an agent of communism for doing this, but it's basically an extension of what his work was with gull wasps, is that if you truly want to understand a living population, whether it's gull wasps or people getting it on, it's not enough to just take a small handful of examples and arbitrarily decide this is the way the world ought to be. You've got to sample the full spread of variation if you want to have any hope of defining, of any kind of definitions there. It's the same approach to gull wasps as it was in human sexuality. And in fact, he's basically a founder of modern human sexuality studies. Again, some of his conclusions have had to be modified, mostly because, again, you have to be very careful about how you are phrasing survey questions and how you administer the surveys. And in some areas, morals and behavior patterns have changed. A lot fewer people in Indiana grow up on farms, so I'm assuming that, sexual experimentation with the sheep is a lot less common than it used to be. One would hope. So yeah, actual conclusions may have shifted, but the basic idea of sampling a huge spectrum of variety and diversity and variation and trying to come up with a science that encompasses all of that and takes it all in and deals with it directly, that is very important in both sexuality studies and in biological classification. You've got to encompass that full-blown spectrum, and Kinsey understood that probably as well as anybody ever has. And it shows up in both his studies of sexual behavior in wasps and of sexual behavior in wasps. All right, that seems like a good place to take a break. I've got 11:27. Tell you what, let's reconvene at 11:40 and I'll flap my gums at you for a bit, and then we can go home and you can all study. So take about 13. I'll stop recording.